very glad and honored to have Gérald Späth here. Gérald, it's a friend for a couple of years that we are trying to work together. Gérald Späth is really one of the most important researchers in the field of Leishmania host. I put the recording. In the field of Leishmania host interaction. He currently holds a research director position at Pasteur Institute in Paris, where he directs the unit Parasitologie moléculaire et signalisation and the department Parasites and Insect Vectors. He and his team are global leaders in analyzing the biology and mechanism of this interaction, Leishmania and host, using several approaches: molecular, genetic, pharmacological, and immunological approaches. Gérald and his team have made seminal contributions to our understanding on how these intracellular parasites adapt to their macrophage host cell through several steps of this interaction, several stages. He combines using really he's creative in this capacity to combine basic results obtained in his investigations and the discovery of new drugs by targeted parasite protein kinases and macrophage epigenetic enzymes. He animates also an international research program through the coordination of large national and global networks, including Leish drug translation and others. And finally, not least important, he animates the Leish Read platform, coordinates funded Leish Healed program, and directs an international mixed unit with a partner team at Pasteur Institute in Shanghai. I've just summarized his curriculum, and again, I thank you, Gérald, to having accepted the invitation. Thank you.
Fantastic. Thank you so much. Thanks, Patricia, for, of course, the invitation, organizing this seminar, and, of course, the very kind introduction. Maybe for the sake of the presentation.
My screen and my if you need something, I'm here.
Okay, yeah. And maybe everybody that is not speaking at that moment, please turn off your microphones like that. There is no background noise, But if you see that the slides are not advancing or I'm not talking anymore, maybe just shout at me, So there might be something wrong on my
side.
Just,
Gérald, to remind them that most of the questions will be done by chat. Thank you very much.
Yes, perfect. And we will go over these at the end of the presentation, of course, Yeah, as I said, it's a pleasure to be here. I would love to be in person, but, this is the best, next best thing that we can do going online. So I'm going to talk about the following project today: going nuclear, Leishmania, epigenetic subversion of macrophage immunometabolic functions. But before I will start with the science, I want to, as usual, wake up the students and maybe also the adults in the audience by just giving you a very short introduction of Leishmania in the more Hollywoodian context, the Alien versus Predator, because if you've seen the movie Alien versus Predator, I would say the Leishmania infection cycle is corresponding reasonably well. Now, in that movie, you have, of course, some protagonists. One is the alien that is shown here. And in our story today, the alien will be the protozoan parasite Leishmania. Now, not only that, like an alien, it has its own spaceship, shown here as these phlebotomine sandflies, but also the biology of the Leishmania, which is a eukaryote, but we have learned over the past few decades that biology is really extraterrestrial, very, very different from other more classical eukaryotes. Then, of course, in the plot, you have the next actor is the predator, showed here. And in our story, the predator is the macrophage that, of course, it's a very important immune cell in your blood and in your tissue that is designed to eat any kind of foreign material and, of course, to ingest microbes and eliminate those, and so to protect your body. And, of course, you may know that the macrophage is kind of a sentinel cell, so it's at the basis of all immunity that signals either danger or infection, or can signal also to shut down immunity if everything is resolved. Now, I wouldn't be here today if the predator will kill the alien. Actually, this is not the case. The alien Leishmania is actually surviving the very toxic macrophage and even exploits the macrophage as a host cell. And here I show you how these parasites develop inside the macrophage into amastigotes. So that's the intracellular form of the parasite that is adapted. And at that stage, the parasite literally takes over possession of the macrophage and modulates these cells for its own survival. And of course, the outcome of that are these immunopathologies that you see, which are really due not to the parasite itself, but to the dysregulation of your immune system in the presence of the parasite, because of course they dysregulate macrophage functions. Now, going back to the real world, Leishmaniasis, the disease caused by Leishmania, is of course a global health threat, as you can see here by this world map that shows the distribution of autochthonous Leishmaniasis in about 100 countries. We distinguish different types of Leishmaniasis, cutaneous, mucocutaneous, and visceral Leishmaniasis, which depend on one hand on the parasite species, and on the other hand also on the immune response to these parasites. Now we have several challenges that our scientific community faces since many years. So we have recurrent epidemics in different parts of the world. We don't really understand, it's often cyclical, so we do not understand really what controls Leishmania infection. We have also noticed an emergence of Leishmaniasis, notably in Europe, in Greece, and also in Southeast Asia, with first cases now shown in Thailand. We see an urbanization of the disease, notably in your country, in Brazil. And of course, the biggest problem that we have is that we see a global rise in parasite drug resistance, which is, of course, in the absence of new drugs and treatments, a big problem. Now, I remind you that there's no human vaccine available against Leishmaniasis or Leishmania. And of course, our current treatments are suboptimal and threatened by drug resistance, as I just said. Now, I want to right away jump into the subject of today. At the basis of Leishmania pathogenesis is, of course, as I said, the infection of professional phagocytes, neutrophils, monocytes, and macrophages. And it's the immune subversion that the parasite causes in these cells that, of course, generates the immunopathologies of the different forms of Leishmaniasis. Now, the parasite is also able to be taken up by so-called non-professional phagocytes, epithelial cells, fibroblasts, and also dendritic cells. And we believe that the parasite kind of nestles down, persists in these immunological less dangerous cells, and thus escapes from immune detection. The uptake of Leishmania, in contrast to other parasites, is passive, so there is no invasion machinery. It's just bluntly targeting the macrophage for phagocytic uptake. Parasites then are inside a phagolysosomal vacuole that acidifies. It's like a digestive vacuole, but Leishmania resists the digestion, even converts into this intracellular amastigote stage. Now, there is a very characteristic development of the disease that we have seen that the first phase of infection occurs silently, so the immune system is not really detecting the presence of the parasite. But then somehow the immune system is aware that there is an infection going on and mounts a very overarching inflammatory response that then causes the tissue destruction. And eventually, if the host is immunocompetent, the parasite can be controlled, enters a chronic phase where the parasite persists for the rest of the life. So the immunity cannot get rid of the parasite. And of course, it can come back if the host is immunocompromised. Now, as I said initially, the different pathologies of Leishmaniasis are intimately linked to the type of immune response that is generated by the parasite. And there is a nice review from Phil Scott that gives you an idea. I'm not going to go over it in detail about the contribution of antibodies or delayed type hypersensitive response and different cytokines to the different forms of Leishmaniasis. Now, the key question that we address and others since many, many years is, how does Leishmania survive in host macrophages inside a highly inflammatory environment? So these lesions that you see here are, of course, caused by inflammation, and they're full of parasites and infected macrophages. So somehow Leishmania has evolved to survive this inflammatory environment. So we, when I talk about we, this is my team. We have about 14 people that are divided in 3 sub-teams that have complementary questions on parasite-host interaction. We have one team that is more about the parasite, how parasite evolves and adapts in the insect and in the macrophage and mammalian host. A second team that asks the question, how is the parasite communicating with the macrophage by the release of exosomes and ectoproteins into the macrophage cytoplasm. And finally, we have a third team that looks at the consequences of infection on the host cell and the host as well. And due to the time reasons, I only have time to talk about that subject today. So here we have the sub-team that is co-managed by me and Eric Prina. And it all started about 5 years ago when we had a nice collaboration with a Chinese colleague at the Pasteur Institute in Shanghai, who is an inflammasome expert. And so we were deciding to collaborate on the question how Leishmania macrophage infection affects the inflammasome of the macrophage, which is a very important molecule for inflammatory responses. Now, there are different types of inflammasome receptors, and we were in particular interested in one called NLRP3, which has been previously linked in other systems to infection. So here is a schematic view of this inflammasome complex that has different parts. There's a caspase 1 that is activated by interacting with the ASC adapter and the NLRP3 receptor molecule, and this whole machine converts pro non-secretable IL-1 beta into the secreted form, which then of course induces the pro-inflammatory response. And so we investigated how Leishmania infection affects this response in different systems. Here we have an in vitro system where we use bone marrow-derived primary macrophages that are infected with Leishmania amazonensis amastigotes that we isolate from a mute mouse. So it's a very physiologically relevant system. We can then visualize what happens inside these cells by different markers. So we're using an M-cherry transgenic parasite that you can see here in red. We can, of course, stain for the host cell nucleus with Höchst. And we also can stain the parasitophorus vacuole that hosts these parasites by using lysotracker green. And finally, we can then go from the in vitro system more towards the in vivo setting, where we infect mice with a parasite, and after a few weeks of infection, when the lesion is growing, we can isolate the infected macrophages. And so these are in situ, in the tissue, infected macrophages, and can test their biology and compare them, of course, to our in vitro setting. Now, to cut a long story very short, because I have so many unpublished results I want to show you, we published in 2020 that Leishmania infection of macrophages completely blocks the inflammasome. First of all, Leishmania itself does not induce the inflammasome, so there's no response of the macrophage. And even worse, if you try to activate the inflammasome in infected macrophages using a combination of LPS as a microbial stimulus and ATP as a danger signal, these signals are not anymore inducing a strong inflammasome response. So we also asked the question, but why are these things not properly expressed anymore? And we could see that actually it's not only the NLRP3, it's all kind of different inflammasomes that are not responding properly anymore. This, I show you here a schematic view of real results. This is an RT-PCR experiment, quantitative RT-PCR of about 50 genes or transcripts. And so what we could show is that at the center of this down modulation of the inflammatory response sits, of course, the usual suspect, NF-kappa B, a transcription factor, that is a key activator of the inflammatory response. What we could show by mapping the expression profile across the entire NF-kappa B pathway, so these are all molecules that control the NF-kappa B activity. We had a very interesting finding in our qPCR experiment that the Leishmania infection causes a downregulation, here indicated by green, of pro-inflammatory actors, but on the other hand, caused an upregulation of many anti-inflammatory actors. So it was a very smart double, I would say, suppression of the NF-kappa B pathway. Now, once we've seen this kind of very fine-grained regulation on the level of the NF-kappa B pathway, we of course asked ourselves, how can this be regulated? What is the basis? Is there maybe epigenetic regulation involved? And so the next question was, how is Leishmania infection affecting the epigenetic profile of the macrophage? We used a very simple assay where, well, first of all, I have to tell you that, of course, most of you may know that the gene expression level can be regulated by chromatin condensation. Condensed chromatin is a closed configuration, so the gene is off. And upon chromatin modification, the chromatin is loosened up, and we say it's open chromatin, which of course can trigger gene expression. And this can be triggered by acetylation, for example, of histone H3, but also of different types of methylation modifications. So we used, in order to monitor acetylation and methylation, the following kit that was provided by QIAGEN, where we used different antibodies against certain epigenetic marks or histone modifications. In this case, we had, and I marked them here, so this is histone H3 with the N-terminal domain where a lot of modifications happen. And so we had antibodies specific for acetylation of the lysine 14 and 9. And we also had antibodies for the methylation of lysine 9 and lysine 4. Here, I just give you an idea of what this modification is doing in terms of gene expression. The acetylation causes an upregulation. The K9 methylation, so here the methylation causes a down modulation of expression. And on the lysine 4, it's the opposite, that methylation plus causes an upregulation of expression. So what we did, we used this antibody to precipitate the chromatin of promoter elements. This was all designed by the kit. The kit comes with PCR primers to amplify the promoter elements of different pro-inflammatory genes. And so, of course, the more amplification product you get, the more modification you have. So here's the result. I want to first just go quickly over the lysine K9 methylation, which is a negative mark. So it doesn't really, it reduces expression. There was not really too much happening in the presence of Leishmania. So you see control Leishmania infections. There were some genes or some promoters that are hypermethylated, which suggests that these genes may be less expressed in the presence of Leishmania. But what we found is that when we looked at the activation mark, which is this lysine 4 trimethylation, in control parasites that are not infected, we had a lot of mark in these pro-inflammatory promoters, and the presence of Leishmania brought this very much down, which of course indicates a shutdown of pro-inflammatory gene expression. We also saw the same here for the acetylation. Again, it's a positive regulatory mark. And so here you see the infected macrophage signal versus the non-infected, and you can see that most of the signals are shifted down of the diagonal, which means they are hypoacetylated, suggesting that Leishmania also reduces the acetylation level and thus the gene expression level. Now we went on to test this not only in vitro, but we used our in vivo, ex vivo system of lesional macrophages. Just as a reminder, we infect mice, we isolate the foot pad, we inject the foot pad with collagenase and others in order to very gently extract out the infected macrophages, which you can imagine, they're full of parasites, so they are very vulnerable. Here's a first picture of these macrophages. They are extremely full of parasites. Parasite here is again tagged with cherry red. And then when we tested the biology, so we put them in culture and immediately tested them for the expression level of certain pro-inflammatory genes, and also, of course, the inflammasome receptors. And as you can see, there is no activation compared to control, which is LPS stimulated. And we also tested the level of the acetylation of histone H3, which you know, should be, if things are expressed, should be up. And again, you can see that these ex vivo macrophages, they have a very low acetylation level, suggesting that all the promoters that are tested here are actually rather shut down. Finally, we also took the ex vivo macrophages and stimulated them with LPS ADP to see if we can produce a inflammasome response. And as measured by the absence of the strong IL-1 beta induction, we could confirm that even in real macrophages in the tissue, they are blocked for inflammasome activation. So that led to a few papers. First of all, of course, this is the paper. Oops, I have here a little message to myself, but it doesn't matter. I will remove this later. So here's a paper we published 2 years ago in Cell Reports that indicates the model where, of course, the parasite modulates the acetylation and methylation that affects NF-kappa B pathway, and this, of course, inhibits the pro-inflammatory response, and this is how the parasite survives. We also published a follow-up review on it's kind of an overview on how we think Leishmania affects the gene expression in the macrophage through the epigenetic regulatory factors and also different sets of transcription factors. So if you're interested, I invite you to read this review. Now, the next idea we had was, of course, so let's go global, let's go genome-wide and perform an RNA-seq analysis, which I show here. It's a quadruplicate experiment, so 4 biological replicates. And here I show you volcano plot where you see the fold change as a function of the p-value. Everything colored is having a significant p-value, and we see massive down and up regulation caused by infection, about 9,000 transcripts that are statistically significant up and down regulated. Of course, we investigated what are the pathways that are affected by the infection, and this is shown here in this pathway analysis. The different pathways are indicated here. In red and green, you see the amount of transcripts in a given pathway that are either downregulated or upregulated. So you again see there's this dichotomy that it's not just simply that Leishmania downregulates things, it really modulates things in 2 ways. And when we had a closer look, it was quite interesting because it could inform us quite well on what actually is the phenotype of Leishmania-infected macrophages. And we even corner the name LIMS, Leishmania-infected macrophages, because you may have heard about TUMS, tumor-associated macrophages, and we said, why don't we just invent a new name for this kind of macrophages that are Leishmania-infected? And so I have here a little color code with these triangles. All the pathways in black are actually somehow linked to cancer, molecular mechanism of cancer, p53 signaling, hepatic fibrosis, or lung cancer signaling. So there is something going on that Leishmania infection causes an upregulation of all kinds of cancer pathways. So cell death is certainly an interesting point to study. Then, of course, we have different immune pathways, including the inflammasome pathway that are affected. And we also had metabolic pathways that are affected, notably the cholesterol biosynthesis. So what I would like to do, and I'm checking the time, okay, what I would like to do now is just going one by one about what we learned in these 3 phenotypic aspects, cancer, immune response, and metabolism in our system, to better define, of course, the phenotype of these limbs. So first of all, based on our RNA-seq results, it seems that the limbs, the Leishmania-infected macrophages, show an anti-cell death expression profile. So we investigated 3 different cell death, regulated cell death pathways: apoptosis, pyroptosis, and necroptosis. Of course, there are a lot of genes associated with these pathways, and what my coworker Hervé did, each pie is, of course, 100% is all the genes that are annotated for apoptosis and so on. And he could see that 64, 76, and 60% of all the transcripts associated with a given form of cell death were modulated in the presence of Leishmania, highly significantly. And here he dissociated this percentage into all the pathway members that are known to be activating, in this case, apoptosis, or inhibition of apoptosis. And again, you can see that there is a lot of things that are upregulated, genes upregulated that are inhibiting apoptosis, and the activators, they are downregulated. So again, we have this dichotomy that I showed you for the inflammatory response. The same is true for the pyroptotic response, and the same for the necroptotic response. And now if we look in the key factors that are associated with these forms of cell death, so you can have this kind of Venn diagram. Most of course, cell death regulators are shared between the different forms of cell death. Notably, there's a few that are really shared and important for all types of cell death. And You know, there's, for example, caspase 8, which is green because it's downregulated in the presence of Leishmania. And then we have these two actors which are upregulated, and they actually inhibit apoptosis. So this is just to demonstrate you again that there are key regulators of all kinds of cell death, which again show this dichotomy in regulation. Now, if you look at the cells, these are now primary bone marrow macrophages that we put in culture. And if you're working with those, you know that they cannot be maintained in culture for a long period of time. And so what we did, we just measured what's the effect of Leishmania infection on the survival in culture. And we could culture bone marrow derived macrophages for up to 50 days or, you know, nearly 2 months in culture. And I guess we can even continue. And here's just a photographic image of the original cells. You see the cell outline with the Hoek staining of the nucleus. Then day 3 infected macrophages, where you already see the red of the parasite, which of course is increasing in amount. And you can also guess the size of the cell of the macrophage is swelling and swelling and swelling. So these parasites can really inhibit apoptosis or cell death in this system. Now we went one step further, and we triggered cell death in order to see whether the presence of Leishmania can also avoid induced cell death. And so in order to investigate apoptosis, we just removed or deprived the cells from CSF1. A bone marrow macrophage needs CSF1 to survive in culture. And in order to trigger pyroptosis, which is inflammasome-dependent death, we just added LPS ADP. Now in uninfected, well, I have to say that the dead cells were stained with UPRO1. We monitored the host cell nuclei with Herx, then the parasite, as usual, is of course the red signal that you see here. In uninfected ones, we had an important amount of dead cells, which of course with the time increased. In the limbs, Leishmania-infected macrophages at day 3 post-infection, there were only very few macrophages undergoing cell death, despite the stimuli we apply, and the same was true for the limbs at day 30. And of course, we quantified this as a function of time. So in our apoptosis system, control parasites die very fast, whereas this is, of course, the percent of dead cells, whereas the limbs, they showed very little cell death, even at day 50 after infection. Same was done with the pyroptosis. So again, we have the UPRO staining. Here we had treated the macrophages, infected and uninfected, with LPS. And then we added ATP to stimulate pyroptosis. And this is just the time after ATP addition. And you can see that there's a very strong and fast pyroptotic response in the control, which is diminished. I would not say it's absent, but it's strongly diminished in the Leishmania-infected macrophages. So this is a small conclusion, intermediate conclusion, where we say, yes, so there is indeed something going on that looks like these macrophages are more or less immortalized, at least in culture. And so the next thing that we addressed was the immune response. So what's the immune status of these infected macrophages? Now, you know that, of course, macrophages are very well known for their phenotypic plasticity. They can adopt different phenotypes according to their function, and we distinguish different polarizations. There's a whole spectrum between what we call M1, which is a pro-inflammatory macrophage, and M2, which is an anti-inflammatory or wound-healing macrophage. And so we were asking the question, what is the polarization profile of these Leishmania-infected macrophages? Because we already know they're anti-inflammatory of everybody in the field, and we too believed, okay, they are just M2. This is enough. Leishmania shifts them to M2. That should be good enough. But we had a little surprise here. So the first thing we did, we were establishing, this is Sheng, a PhD student from China. Sheng established protocols in order to derive from M0 bone marrow derived macrophages M1 by stimulating with LPS interferon gamma, and M2 by stimulating with IL-4, IL-13. And then, of course, he was comparing the M0, M1, M2 to the limbs Leishmania-infected macrophages. And first, he was mapping out the expression profile of M1, M2 markers, and generally inflammation markers by RT-qPCR, which is shown here. So up here, you see all the different transcripts he was monitoring in M2, M1, and Leishmania-infected macrophages. And we have different groups. So you have a group where the limbs, they look like M1. You can see the pattern is pretty similar. Then we have a group of transcripts where the limbs, they look like M2. Of course, you see here, this profile up and low is similar. And then we had some transcripts where limbs seem to be very specific because the M1, M2 didn't look like the limbs at all. And here I just show you a few of the markers in a more detailed fashion, where we can say that, for example, here there is an M1 marker, IKKA, which this corresponds to the limbs as well, even if it's an M1 marker. We have, for example, here let me see, the IL-1R1, which is an M2 marker, which is also up in the limbs. And then, of course, we have other things that are not even detectable in M1, M2, which are upregulated in limbs. So this seems to be quite specific, parasite-specific response. So this was on RNA level. We went one step further, and we used this proteome profiler cytokine array, which is a very simple thing that you have a capture antibody spotted on a membrane. This is, of course, for many different cytokines on the same membrane. These antibodies will capture a certain cytokine or chemokine that you then can detect and visualize by this enzymatic assay. And so using this membranes, we were monitoring and mapping out the chemokine cytokine profile of our limbs. And again, we have the different groups that I showed you before. For example, if we start here, we have the limbs that look like M1 for these cytokines. We have then the limbs here outer that looks like the M2 for the other cytokines. And then we have one group where the limbs show a very unique cytokine profile. This is also shown by this violin plot here, that the limbs are not really corresponding neither to M1 nor to M2, which we believe is quite an important finding because the field today really thinks that the Leishmania-infected macrophages are just M2-like. This is not the case. So I want to conclude here this part, just to remind you that the data that I showed hopefully convince you that we have a quite unique phenotype and polarization profile in our limbs. So the next step in the next step was then to assess the metabolism. Now, it is very well characterized that the M1, M2 dichotomy of pro-inflammatory, anti-inflammatory macrophages is linked to a very specific metabolism in these two different polarized macrophages. Notably, the energy metabolism is very different. So the M1 undergo glycolytic energy production, so use glucose, and in the M2 we have more oxidative phosphorylation that occurs in the mitochondria as the main source of ADP production. So we decided to ask the question, are limbs also showing a mixed metabolic profile like we've seen on the expression? We use this machine called a bioenergetic flux analysis, also CHORS. It's a quite neat machine that allows you in real time to probe how much glycolysis is going on by measuring acidification, because the glycolysis leads to lactate secretion, which can be measured as a drop in pH. And it simultaneously measures respiration, which means the oxidative phosphorylation, which is oxidant specific or dependent. So you will have in respiration a drop in the oxidative or the oxidant levels in your well that you see. And so this machine is based on this kind of insert that goes into your 96 well plate. This is also shown down here. You see all these nozzles that will go down. There is a light coming through these because at the end you have two probes, like fluorophore probes that are embedded in the plastic, which will be excited by the light. And there is one probe that is specific in order to detect the level of oxygen, and the other one that is specific to detect the level of H+ or the pH detection. This system also has two channels that allows you to put in drugs and metabolites during your measurement, which of course we did as well. Again, this was a very long, complicated procedure, but finally we, Sheng, I have to say, really got the hand of it. And here is just a very simple, you know, there's a lot of different plots that come out of this, but I just want to keep it simple for you and me. So on one side, we can see the glycolysis activity as measured by lactate secretion and acidification. On the other side, we see the oxygen consumption, which of course is a sign of oxidative phosphorylation as ATP production. So the level globally, both parts, if you add them up, will be globally the level of ATP production of your cell. So what we can say is if we see the M0 macrophages, they're a little bit producing less ATP than the polarized, which makes sense because they're quiescent. If we compare now our limbs in terms of energy production, we see that they have a minor increase compared to the M0, a minor increase in glycolytic activity, which is similar to M1. And they have also an increase and maintain a strong component of the OXPHOS, which makes them more M2. So as a matter of fact, they are actually showing something that is more in the middle. The overall production is not significantly changed if you compare it to the other polarized macrophages. But when we look back in our RNA-seq experiments, we said this doesn't make really sense because one of the strongest findings in our RNA-seq experiment in terms of metabolism is shown here. I have a KEC map shown here where we have the expression levels between M0 and LIMS. With everything that is in red are enzymes whose RNA is more abundant in the presence of the parasite, and in green you have a reduced abundance. This is the KEC map of the glycolysis pathway, and many enzymes are actually induced in expression during Leishmania infection. So it didn't really make sense that we have this very limited glycolytic activity compared to the expression pattern. So we decided to use a drug which is the L-leucine methyl ester, which has been published a long time ago as an extremely efficient drug that can kill Leishmania ambutonensis. In a matter of an hour, you have no parasites anymore. So we applied that in order to remove the parasite from the limbs. And lo and behold, while the loiomi, we call it loiomi short, did not really affect any of the other readouts significantly, there was a huge important shift in the limbs where we got rid of the parasite, that somehow now the glycolytic activity was all the way up, really reflecting the expression pattern. So somehow Leishmania seems to limit the glycolytic activity of its host cell, which makes sense because glycolysis is linked to antimicrobial activities. And so maybe it's an immune subversion strategy of Leishmania to limit the glycolytic activity at all. And so to give you the last conclusion, and I have maybe another 5-6 more minutes, this overall conclusion, we believe that so Leishmania modulates gene expression and epigenetic profile that causes a change in cell survival. The macrophage doesn't die anymore. There is a modulation of cytokine secretion and of course a modulation of energy production. And all together, if you integrate all of these signals, we believe that everything that I showed you establishes good conditions for internal Leishmania survival, but also external, because the macrophage, of course, in reality sits in a tissue and communicates to its environment. And so both the different modifications and modulations seem to be designed to allow Leishmania to survive optimally. So what do we do next? Of course, we want to study all of these things, cell survival, cytokine secretion, and energy production in our ex vivo system that I showed you before for the epigenetics. So isolate macrophages from the food pad. We're going to use a very interesting mouse model called mafia mice. This is a good colleague of mine from Pasteur, Philippe Bousseau, who has them. These are mice and macrophages that allow you to induce apoptosis. And we want to study, of course, the anti-apoptotic effect of Leishmania in the mouse, for example, by ear infection in these mice. And then we are undertaking more mechanistic analysis, for example, ChIP-seq, in order to map a genome-wide the epigenetic profile. We're going to use pharmacological inhibitors, notably for the energy pathway. We're going to perform host cell genetics, so lentiviral overexpression or CRISPR-Cas knockout. We're going to investigate the nuclear proteome and also microRNA expression. And we have, I'm not going to go, I don't have time to go through that. We have very interesting preliminary results for most of the mechanistic analysis. We have seen massive changes of the nuclear proteome in the presence of Leishmania. We have also seen accumulation of proteins from Leishmania in the macrophage nucleus, and they're all nuclear pore complexes and nuclear envelope proteins, even histones. So these are Leishmania proteins that we find in the macrophage nucleus, so you can imagine. And also we have done now an RNA-seq on the level of microRNAs, and we could really map the overexpression of certain microRNAs to the inhibition of certain target RNAs in Leishmania-infected macrophages. Basically, I want to do a little switch here for the last few minutes, just show you what we do in terms of translational research. Of course, you already know this picture where we say Leishmania modulates the cell, very much so. And we had the idea in this review that we published in 2019, maybe we can actually interfere with what Leishmania is doing to the macrophage. And the question here is, can we target the host cell epigenome, epigenetic regulation, to cure the macrophage, you know, basically to inhibit everything that Leishmania is doing. Now, this strategy of host-directed therapy is a very interesting one, notably because Leishmania is prone to develop drug resistance phenotypes. And so in my other life, and this is something that Patricia was saying already, I'm coordinating big consortia across the different Pasteur institutes, and this is one where we investigate Leishmania evolutionary adaptation also to drugs. So we published a lot of papers, and as a matter of fact, we have now a model where Leishmania uses intrinsic genome instability, amplification, deletion, and so on, in order to constantly generate phenotypic variability, and ultimately that leads to fitness gain. And so we believe that no matter what drug you develop that kills Leishmania directly, there will be a selection for drug resistance. And so if you kill Leishmania in an indirect way, by host-directed therapy, we believe that this will be less prone for drug resistance. And so what we did a few years ago, this is Suzanne, that was a PhD student a few years ago, and now she's followed up by Maria. We use our macrophage system that you know already with the fluorescent markers, where we can monitor the host cell nuclei, the vacuoles, and the parasite, and we perform phenotypic drug screening. Here we used a highly selected inhibitor library of different epigenetic inhibitors that target all kinds of epigenetic families, regulatory families. And what we've seen is that, well, most of our compounds that are denoted here by these dots were not active because this is a parasite survival. They were all scattered around 100%, but we had a few hits in this epigenetic inhibitor library that were quite selective and strong against Leishmania. Now, this is just another way to show you the same results. This is the drug screening that we do in intracellular mastigotes and using the macrophages. So this is, here we plot the parasite intracellular survival in the presence of these compounds. Here we plot the extracellular survival because we screen also on the free parasites. And of course, if both are hit, you find the compound here. But what was really interesting was a compound class we see here that did not have any effect on the survival of the macrophage, but they very efficiently killed intracellular mastigotes. So this is the definition of a host-dependent hit. We were looking into these compounds, and as a matter of fact, there were several compounds that were very well characterized already, and they were shown to actually inhibit mammalian histone-modifying enzymes. Another hint that this is indeed the macrophage is the target. And for most of these enzymes, there is no homolog in Leishmania anyways. We are following up on one inhibitor, which is an LSD1 inhibitor, shown here. Don't want to go into detail, just tell you that the LSD1 is an demethylase that removes methylation from the lysine 4 and lysine 9. It's also a very important scaffolding protein on promoters, so it allows assembly of different kind of complexes. This has been, this LSD1 molecule is actively pursued in pharmacology for drug development against different types of cancers. And we have now for the first time used these two cancer drugs that are developed currently, and we have shown that we can repurpose them actually to treat intracellular Leishmania infection. So I'm going to go very fast because there are only a few more slides left, but maybe it's a little bit too much for you guys. What we want interested now is, of course, to understand what is the drug, how is the drug eliminating the parasite. And so we did an RNA-seq experiment of non-infected macrophages and infected macrophages, and also infected, non-infected in the presence of the drug, just to see. Here again, this is just a comparison I showed you before, infected versus non-infected, lots of differential expression, and we can map that most of what happens is that the inhibition affects the antimicrobial response of the macrophage. That's what Leishmania is doing. Now, if we compare the two different RNA-seq experiments we did here, the infected versus non-infected, so these are the changes caused by infection. If we now compare this with an infected macrophage that has been treated, we can actually see what actually is the LSD1 inhibitor treatment reverting back to normal. And so these are two data sets which I combined in a very fancy visualization. So the differential expression that you see here, I was projecting first the effect of the parasite infection is projected as an outer ring. Green is downregulation, or red will be upregulation. And I did the same for the inhibitor-treated infected macrophages compared to the non-inhibitor treated, but it is the bubble, the color of the bubble that indicates the direction of the expression. And so what was really intriguing is these are now all the most important differential expression changes. You can see that all of these transcripts, most of them have a kind of a how can I say, opposite coloring. When you have a red ring, which means Leishmania infection induces the transcript, you have a green bubble in the middle, which means if you add the inhibitor, LSD1 inhibitor, that transcript is actually going down again in expression. And we had the opposite down here. You see the green ring, which means these are target genes that are downregulated in Leishmania-infected macrophages, but they are rescued by the presence of the LSD1 inhibitor. And so in over 80% of all Leishmania-modulated genes were actually reverted to normal by the presence of this LSD1 inhibitor. This is just, again, a pathway mapping what the LSD1 inhibitor rescues, and you see defense of virus response, interleukin-6, inflammatory response. So the antimicrobial response is rescued by this inhibitor. And so here's my last model, where we believe that, of course, Leishmania somehow activates and interacts with LSD1 in order to reprogram the epigenetic profile of the macrophage. And of course, once we add the inhibitor, this reprogramming is not going well anymore. And so most likely that causes Leishmania death. One hint that we have, LSD1 is not changing in expression, neither on the RNA nor on the protein level, but we use something that is called a cellular thermal shift assay, a CETSA, so these are extracts that we obtain from infected macrophages at different they were treated with different temperatures where proteins start to, of course, aggregate and fall out after centrifugation. And here we measure how long can a protein be soluble at high temperature, because if a protein is interacting with something like a drug or another interaction partner, it is actually more soluble and is not centrifuged away. Here's a Western blot of these different extracts, just revealing LSD1 itself. And you see that we lose LSD1 at about 46, 49 degree temperature, treatment of these extracts, but it's stabilized for much, much longer in the presence of Leishmania. So the presence of Leishmania stabilizes LSD1 in this assay. It's also shown here, indicating that most likely how LSD1 is interacting is very different in the presence of Leishmania compared to non-infected macrophages. And I want to jump here, and I want to just thank you for your attention. I'm looking at the time. Okay. So this is my wonderful team. These are my wonderful collaboration partners for that project. So we have Antonello Mai from La Sapienza University in Rome that gave us the initial compounds for the drug screening. And now we have a big project with Manfred Jung, a chemical epigeneticist or an epigenetic chemist from the University of Freiburg. And we have a nice project that is just funded now. And I thank you very much for your attention. I stop here. I go out if I can.
Thank you very much, Gérald. Really an amazing presentation with the integrative analysis of this very complex interaction between Leishmania amazonesis and host cells. Probably you have also in other Leishmania species. We have Mr. Maya that put his hand on in the middle of your presentation, so I have to to ask him to. Oh, he's here. Okay, Mite, please.
Hello.
You have some
Thank
questions
you.
also. Okay.
Thank you very much. It was really very impressive presentation. I have three questions.
Yes, please.
First of all, have you used human macrophage? Probably I missed, or you are using only mouse macrophages?
That's a very important question. So we are using human. Yeah, let me answer the question right away. I will not remember the first question when you. Yes, we have used PBMCs in the context of our drug discovery project, the last part that I showed you with this LSD1 inhibitor, which we confirmed that this inhibitor is working very well as well. We did not yet include it in the validation step of all the LIMS phenotypic analysis, which of course you're totally right. This is something absolutely essential. We don't want to study the mouse. We want to learn something about the human. So I appreciate your comment. Definitely we're going to follow up on PBMCs.
My second point, have you looked if zinc and vitaminas play games in the epigenetic regulation factors? Since they are really important.
Hang on.
Zinc and vitaminas.
Zinc. Oh, sorry. Zinc. Yeah. Sure.
Zinc and vitamin, if they play games, since they are really important to do in the folder or the finger protein that regulate expression.
Very interesting.
My last question.
Yes.
Since infected macrophage, since Leishmania is able to download the anti-death molecules, okay?
Yeah.
Do you think this can, in the very end, helps for skin cancer development in people that has Leishmania for a long period not treated?
That's a very interesting question. Of course, we're far from that investigation. In my humble knowledge of the literature, I do not see any kind of higher incidence of, I never read a paper that showed higher incidence of cancer, skin cancer, for example. I did not.
Our friend here, Gabriel Grimaldi, long time ago, he called attention for that.
Oh, really? Tell me, what was the result?
Gabriel Grimaldi, which probably
researcher at Fiocruz Bahia.
Yeah.
Great.
He was from Rio in this time, no?
Yeah. He's from Bahia, then went to Rio de Janeiro, he went to France, and then came back to Brazil, to Rio de Janeiro, and now in Bahia. Thank you very much. I end my questions.
Perfect. So for the first question, of course, we will definitely include the PBMCs in order to validate what we see. Of course, it remains still an experimental system. Even PBMCs are not really reflecting anything that can be seen by a Leishmania and a natural infection. Ultimately, as part of my coordination action with the Réseau Pasteur, we have partners at the Pasteur Institute in Iran who are doing tissue single-cell transcriptomics as part of our project. That will be a real validation step. So we know what we were looking for. If we can confirm it, okay. If not, it means the systems are different. But this is the system that we feel would be the strongest one. Vitamins, it's a very important consideration, notably when we talk about metabolism. I have to admit, we have not yet discussed about investigating vitamins, but I noted it down and it's an appreciated comment. So in terms of cell death, again, I don't have the paper that you're referring to maybe, or the finding. I would appreciate if you could share that with me. I would be very, very interested. That's for sure. Absolutely.
Thank you, Mite.
Thank you very much.
Mite, you have to go on. Thank you very much for your questions. And now we have Leishmaniasis here
So if you may allow me,
several questions from our...
Yes, maybe Patricia, I will just read them like that.
How do you prefer?
Yes, I read them. I'd say the name.
Please. The first one is Camila Indiani.
Yeah, Camila Indiani. So, hi, Gérald. Thanks for the presentation. 2 questions. How do you reconcile the results on L. amazonensis not inducing NLPAR3 activation versus this from Zamboni lab showing the opposite? This is a very essential question. I fully agree.
I had the same question.
Yeah, sure, of course. In the limbs, do parasites replicate and ever burst out of the macrophage? How does it work? That's a very important question as well. So in this cell report paper, of course, we very much discuss the effect of the Zamboni paper. Now, I think that number one, they used promastigotes from culture. We used the lesion-derived amastigotes. And as a matter of fact, in our paper, we also, this was all in supplementary data, we used promastigotes as well, and they were behaving very different. They did not interfere with the inflammasome, but after 3 days when they converted into amastigotes, then they interfered. So if you use promastigotes and you immediately add LPS ADP, you don't see an interference. If you wait for 3 days until the amastigotes have developed, or if you use amastigotes right from the beginning, you will have this observation that I showed you. So we believe that both studies are complementary. One studied more the promastigote interaction with the macrophage, and in our case, we are more studying the chronic phase of the infection, I would say, with the amastigotes. Now, the next part of the question is, do parasites replicate and ever burst out of the macrophage? So, I didn't put all the data, there's so many more data. So we have a whole curve of the time versus parasite burden, and after 50 days, they still accumulate more parasites, you know. So we did not really see anything bursting out, but of course, this is something that may happen in a split second, you know. So this requires, I would say, a much more sophisticated setup than what we showed. Maybe some live imaging over long term to really capture the moment. I don't know if there's really something like that of a bursting macrophage, because you read in many reviews, it's written like that, and then Leishmania bursts out of the macrophage. There is not a single paper showing any burst in my, again, humble knowledge of the literature, you know. We cannot, of course, there are other ways. For example, I showed you that pyroptosis is delayed and diminished, but it may occur. And we've published last year or 2 years ago a paper in journal Cell Science where when we strongly induce pyroptosis, then the macrophages die, even slower than control. Of course, it's the same thing. But then suddenly the parasites are expelled. They are expelled from the dying macrophages. And so I think more than exploding, I would say that there's one moment coming that either pyroptosis will come in play, or maybe the parasite fields, because there must be some kind of quorum effect. Like if there's too many parasites, they cannot all feed on the same cell. Maybe they even trigger themselves at a later stage, apoptosis in the macrophage. The first stage is anti-apoptotic, and the later infection stage could be even triggering, saying like, it's time to leave that empty shell. Let me get out and infect new macrophages. We did not see that yet, but of course this could be because we're in culture. As I said, we're going to go now in vivo in order to monitor all of these things, and hopefully maybe we see some free parasites floating around in the ear that could come from such an event. Then we have Claudia.
Claudia?
Yeah, I skipped the compliments. Limbs have very particular characteristics. Do you have any idea about the alterations in the metabolism of macrophage using other Leishmania, such as L. major and L. brasiliensis? Well, we did not do these studies. I have to say this is maybe one of the biggest plagues of Leishmania research, is that the systems are so dynamic that you can use the same Leishmania species and you use the same black 6 mice, but they're coming from a different company and they were feeding on a different food, so that the intestinal microbiota is different. You will have a different response of the macrophages. This has been published by Dan Littman 20 or 15 years ago, which is crazy, that you think that, we reproduce the results. you cannot, it's really difficult to reproduce. And so I would say, yes, there are, of course, the similar studies using the seahorse setup, the bioenergetic profiling in Leishmania major, notably, there is not so long ago. And I think it was also Leishmania-infected. But they're using different systems, different setups, different timelines, different infection protocols. The only thing I can tell you is that the oxidative phosphorylation was shown in Leishmania major infection also to be promoted more by Leishmania than the glycolysis. But the same study was much more fine-grained, and they said in the very initial 8 hours, it's the glycolytic activity that goes up, and then this goes down and oxphos comes up. So if you look in the literature, there are things that are published which pretty much reflect in our findings as well. So hang on, let me see. Okay. Then we have Lucas. Do you see the same phenomenon in Aldonovani infection? Is this phenotype cytokine dependent? Good questions.
He put something I'm talking about NALP3 and
Hang on,
phenotypes.
I'm at the question. Hang on, this is the question of Lucas.
Yeah,
So
he complement. Yeah.
Yes. So we have done Aldonovani studies. Notably, I don't remember so well, but I think it's also in the Cell Reports paper from 2020 in supplementary data. So we repeated stuff with Aldonovani, and we've seen the same phenomenon on the Inflammasome. Yeah, absolutely. If the phenotype is cytokine dependent, very interesting question, because we also speculated that maybe we have some form of an autogreen loop, that maybe some cytokine is released that is then recognized by the limbs itself. But we did not yet really see any kind of, feedback loop, cytokine feedback loop in our system.
I'm sorry, Gérald. How can you explain this difference? Really amazing how Zamboni, it's completely different results from yours. And can you go further in this discussion, because it would be.
But I already explained this in the first question. No, how is the Zamboni result different? I mean, we're just using promastigote versus amastigote. It's very different.
Do you think that's just okay?
Honestly, I mean, it's in the paper that when we use promastigotes, we actually reproduce what Zamboni was seeing in terms of activation, but amastigotes, it's just not the same.
Okay. I'm not sure, but I think that he tried also amastigote, but I have, maybe I'm
not
wrong.
Published, because we were looking very carefully at that paper.
Okay. Okay. Absolutely. And then you have.
So there is another question of Claudio.
Claudio.
Yes. How is the lipid metabolism in limbs? Did you observe any changes? Oh, yes, absolutely. This is actually a very important consideration. I jumped a little bit over that because I was running out of time. So one of the strongest signals that we get in limbs that was shown in the pathway overview was cholesterol biosynthesis. We have actually an upregulation of all kinds of enzymes that are in the sterol biosynthetic pathway in cholesterol biosynthesis. We even kind of trace this back. There is a key regulatory transcription factor, SRBBP2, which is a master regulator of the cholesterol biosynthetic pathway. So it regulates all the enzymes of the pathway. And this transcription factor, plus all the downstream regulated genes, is upregulated significantly during Leishmania infection, which means Leishmania induces cholesterol synthesis, which is a very well-known fact. It's published by many people. They didn't really look into the transcription factor aspect. We believe that this is important for Leishmania, for all the membranes that they need to build, the phagolysosomal membrane, their own membranes. They depend on cholesterols from the host as well. Now, what I didn't show you is when we add the LSD1 inhibitor, so that's the epigenetic inhibitor that reverts. Again, one of the strongest reversions happens on the cholesterol biosynthesis. Whatever was induced by Leishmania is actually reduced to normal in the presence of the epigenetic inhibitor. We believe that this also could contribute to the Leishmania killing, because suddenly they are in the middle of a growth period and suddenly they are starved of essential lipids that, of course, could have a bad phenotype. for infection. So this was, hang on, Lucas, thanks. Do you have the same feeling? Then Lucas said, I'm talking about the NLP3 and glycolysis phenotype. We did not yet do Aldonovani. It's exclusively done with Inflammasome in that system. I do believe to remember that there may be some groups that have used Aldonovani, you know, with the results that I showed before, or indicated before, that Oxphos is, you know, Oxphos is also making sense because it reflects an M2-like polarization profile, which still I believe that Leishmania is having a very mixed profile, but it still exploits very much the M2 part as well, notably in terms of energy production. So thank you.
Fantastic.
Thank you.
I have a question
Yes.
for you. That's amazing that targeting cancer, you can control Leishmania infection. That's amazing. And methalphosine, it's an example that it's a very good anti-Leishmania that has been used in the last years against Leishmania infection, tegmentary, and visceral disease. We have also found in a proteomic study HSP9 modulated in Leishmania infection, and we tried anti-cancer that target HSP9, and we have fantastic results in vitro, in vivo, and probably going to translation this for a future treatment. How can we explain this in your complicated and fantastic integrative analysis, this type of, how can we explain how Leishmania does that?
Hang on, there was a little glitch. Can you just repeat the last part?
No, I would like you to try us to explain how Leishmania does that to the cell, to be in your integrative analysis. You have the chance to try to answer these interesting questions.
We only scratch the surface, even though you feel there's a lot of data, and I believe we only scratched the surface. And this was, I went very fast over that because of time. This was our follow-up studies, the microRNA. What happens on the microRNA is just mind-blowing. So the Leishmania infection, and this is again Leishmania-infected at this moment, we mapped the microRNA expression pattern of the infected, non-infected macrophages, and there are important differences. Some microRNAs are 10, 20-fold upregulated. And of course, when you have a microRNA that goes up, it kind of brings down the target mRNAs. And this matches with our total transcriptome. We can really see, yes, they're really down, and the microRNAs are up that are known to regulate these things. You know, so this is a new layer that we added. Not only have we transcription factors that are controlling histone-modifying enzymes, but the histone-modifying enzymes are controlling the transcription factor expression as well. So it is such an integrated relationship that I believe the only way we can explain what happens is what we call, emergent property. It's called an emergent property. Emergent property. An emergent property is that the parts, a system that is consisting of different parts is so much more complex than each individual part. There's something through the interaction of all these layers that allows a phenotype to emerge, which we cannot understand because we do not know the individual interaction of the thousands of molecules. There may be even layers of biology we don't even know yet, you know? Circular RNAs are just coming up. This is a crazy thing. So, and so for me, it is an emergent property, saying like, if we down to the quantum level can really understand what's going on, a super intelligent artificial intelligence may give us an answer, which could be 42, as a famous book did, you know, which we would maybe not even understand. But it's for me an emergent property, a self-organizing principle where all these layers are somehow working together. I do not think that there is a single individual factor of Leishmania that is doing this to one factor in the macrophage and then everything is. I think it's a fine tuning. It's always there is something of an equilibrium that is found. Maybe there's a period of things going a little crazy. Of course, every infection, many parasites are not establishing infection. What you see during infection is only this is a successful one. So there is a bottleneck, and you know, you always look at the successful. We should also study the macrophages that are not successful infected. That would be actually the interesting thing to understand. Why is Leishmania not surviving in 20% of macrophages in our culture dish? Is this because the macrophages are slightly different, or there is some balance that suddenly is tilted to an anti-Leishmanial response, you know? So these are questions, but I cannot give you an answer of that. It's just that you know, it's really, really complex. But you know, we're gonna.
Fantastic. Your presentation was really amazing. Thank you very much. And a beauty of your model that I think that it's very important. I would like to emphasize that because you pick up the macrophages from lesion. That is, I think that it's really different from that use model that we cultivate the parasites.
Yes, of course. Thank you so much. I didn't even think about it.
Really, I was wondering that was, and I know that is the macrophages that are in the lesion, so they are different from those that we cultivate.
You know, it doesn't mean that there may be other cells that can mount an inflammasome response, bystander cells. Of course, there is a lot of IL-1 beta produced in a Leishmania lesion.
Absolutely.
But I don't think it's the macrophage that is
I think that this point is very important.
Of course, absolutely. Bystander cells play important role.
Okay, thank you very much.
Thank you so much. This was fun.
Finally being in Salvador. I hope next time you'll be here.
Oh, I come tomorrow. I even pay myself.
Oh, really? So we organize a meeting in innate immune response, autophagy, another. So I will invite you the next time.
That is so nice. I would
love to be very glad to have you here.
Thank you so much, everybody.
Thank you very much.
Good day.
Have a good day. For last of the day, no, you were.
Yes, we're going towards the end. Bye. 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 Bye.
Thank you.
Thank you.